Our first speaker is Huaijun Zhou, who was a PhD student and a postdoc with Sue, right? And is now at UC Davis. So Huaijun, the floor is yours.
Good morning, everyone. Yeah, welcome to the Iowa State. So yeah, it's a great pleasure to be the first speaker. I have lots of pressure. Didn't sleep very well last night, but it seems like Jack gave me extra 7 to 8 minutes because he knows I speak very slow many of the time with broken English. It's glad to come to Ames, Iowa, and even I visited many times yesterday. It's a beautiful day. I have a really great time to visit campus, visit QD. So really enjoyed the Ames, Iowa, which I spend most important of my time here. So what I like to share today, I like to start one very important email, which happened, 27 years ago. So many of you guys probably remember that time our email from this called unverified, but someone accepted it. So this is the email I sent to Sue 27 years ago. And so last year, Sue print out this, find that email and bring it to me. It's a really appreciative of the gift to me and realize the time go very fast. So she accepted me as a visiting scholar to her lab. So kind. If you guys look at that email, it's really broken English. So when I read this, I could, this is my English, but still today, with some improvement, I'm continuing to make effort to improve my English. And then a year later, so she accepted me as a graduate student, GSR code. So that's how I started my many years of the work at SUSNAP. And then you can see I worked pretty hard. So and at that time still looks pretty young. Even nowadays I have more of gray hairs. And then if you look at here, so when I work in the lab many of the times with this black hair, now it's become gray. So you can see time flies. And so I'm not just work as a PhD student, but at that time, so really allows me explore other area, which is bioinformatics and computational biology in a master degree. So I remember I talked to Sue 3 times to ask her if I can pursue a master degree in bioinformatics. Sue said, no, don't talk this time. Come to next time. Then 2 weeks later, I came to her office. I talked to her. She said, "Are you serious?" I said, "Yes." I said, "No, we don't talk about today. Come next time." 1 month later, and then I stopped by her office, and I said, "So I really want to pursue a master degree in bioinformatics." She said, "Oh, okay. Now you convinced me, so I will allow you to pursue a concurrent master degree in bioinformatics, because this is how generous and nice of the mentor, allow me utilize the funding to provide me to doing another interesting in bioinformatics. So I really think how Sue did for me. Obviously, a few years later, fortunately, I was able to graduate with my PhD. So that's time. So basically, give me that, hope. So I formally graduated. And then 1 year later, so allow me as a postdoc work with her. And that's the time exactly 20 years ago. So in 2004, so Sue hosted that farewell party at her house. So you can see, you know, our family over there. So even my son now totally different. It looks so, so this is the chicken. When we chicken stay in the nest for a long time, so that's the time we need to left the nest. So I went to the different place. Since even I left, you know, the nest, we still have many of those contacts, collaboration for many years. So so you can see we in the past many years we have many great memory. So we're not just you know, doing the research, but we do enjoy the mealtime, fun time together. And this is in Africa. And this is the last year when Sue and Greg visit UC Davis. So we had a wonderful time with my training at Davis. And obviously, there's so many of the memories. I just couldn't count. There's so many things I cherish the time with Sue, and so this is many of the wonderful times. So that's the time, you know, we lost actually the African project. We together, you know, left from this airstrip called airstrip. So very short distance for those small airplane take off, you know, from those safari trip. Obviously, it's a great honor, you know, last year, so I received such of the honor, you know, from National Academy of Science. I very fortunately, Sue was able to go with me to receive that honor. So without her mentorship and all of the professional and personal level of guidance, there is no way I can you know, achieve such of, and this year also as a top effort for the Pioneer by World Food Prize Foundation. So this is a huge achievement. So I definitely indebted to what Sue have done for me. I guess she has done so many of impact for many of the people who will probably share their testimony about Sue. So Sue, indeed, the best mentor in the world. And I only can speak very few of the attributes of her mentor style: dream big, forward thinking, inspire others, encouragement, and be positive. There's probably many of the attributes I missed. I'm sure so many of my academic brother and sister will share their personal interaction with Sue, how Sue have the impact for them. So I'd like to spend probably next, you know, gosh, I still have a minute time to talk about the research work, which basically you know, how, you know, my training at Iowa State and SUSNAP can, you know, basically help my research program to expand to many different areas. So I like to start with some of the background as training in the portrait genetics, genomics, and bioinformatics. And then talk about a few of the projects. Start with that USAID funded called Feed Future Innovation App, Genomics to Improve Portrait, primary focus on the portrait disease called the Newcastle disease virus, and then extended to the work called functional annotation animal genome, which expand of the research work not to my most favorable species in chicken, but also to the pigs and the cattle, and also work with other complex traits like growth rate, you know, feed conversion. And then lastly, I like to touch one of the new project we started last year, which collaborated with Hainan International. I know many of my colleagues here. I'm really glad to have this great opportunity to work on this special project focused on the animal welfare. So you can see my interest in continue to explore is so always encourage us to open your eye, open your mind to seeking the world. And then finally, I like to provide some perspective about the research in the genomics and genetics. So as an animal scientist, we are living this world. I guess you guys probably see these. So the world population will increase, about 3 billion in this continent. And then if you look at it here, so you can see most of the population increase in next decades will be from African continent and a South Asian country. If you guys think about this, this is the most of those poor of the countries in the world. As an animal scientist, basically, it's have like 1/3 more than mouse needed to feed in 2050. So this is a great, burden for the scientists working in the animal science, how we can make this effort to feed the people in the world. So this is the world we are living. If you look at this world map, so you can see the different color indicated that if we go this way, so you can see if it's orange, if the red, which means the hunger, the chronic hunger have a higher percentage. So if you look at it here again, so most of this, many of the African countries, South Asian countries, so these have chronic hungers. And then you can see if these trends continue like this by 2030, even like this, we'll have more than 840 million people will have chronic hungers. So this is very important to realize. Many of the time we are live in this beautiful country, right? So we never worry about if we have get the breakfast, lunch, and dinners. But many people in the world, particularly children, like more than 1/4 of those children are stunning and were not able to reach their physical and congenital potential because of they don't have not just the food, but also the nutrition. So that's how it's have more than 2 million of the people worldwide are deficient in those key micronutrients, because those micronutrients are very important for the brain development. So eggs and poultry meat are very important to provide the essential micronutrients. I'm not trying to let the people work in other species like pig, cattle, sheep, offended chicken. Indeed, these eggs, it's most high dense of those nutrients, particular micronutrients for the children development. And then you can see, there's 1/4 of those deaths, all of the deaths is from a non-communicable disease under age of the 60. So you can see nutrition do play a very important role to improve the human health. So for the livestock, really can contribute more than 40% of total value of the agricultural output, and also supports really livelihoods and food security. So that's the kind of the projection talk about by 2050, if we want to feed of the population growth in the world, how much of the increase from livestock, so we can feed the people. You can see it's not like unbiased on chicken. So you can see indeed chicken egg, it's in the top, need 120% of the increase, egg 65%. Other livestock also need to have significant improvement. So that's how the innovation, new technology, so we can be a more efficient and more susceptible to improve the animal production in the world. So I'd like to start with the first project So Sue and Jack be part of this program. So we work together called Feed the Future Innovation Lab, Genomics to Improve Poultry, funded by the U.S. Agency for International Development. So specifically, it's to improve food security in Africa by enhanced resistance to the Newcastle disease virus and heat stress in chicken. So that's a nice picture we have with the team and advisory board for the program. And this is the really multidisciplinary of the research team. As you guys can see, it's cover the topic from genetic genomics, breeding, quantitative genetics, statistical genetics, genetic diversity, poultry virology, epidemiology, international development, agricultural economics. So really a great team with multidisciplines, so we are able to address those challenging issues in the African countries. So obviously the UC Davis and Heine collaborate with a scientist here at Iowa State and then the University of Ghana in West Africa and Sokoine University of Agriculture in the Tanzania, in the East Africa, and also International Livestock Research Institute in Nairobi. So why poultry, I guess, so I probably don't need to spend much of time talking about how important of the poultry for the food security, but particularly for the African country, this is a really important poultry can accessible to the smallholder producers, and then they just need a very small space. So the chicken just running around the chicken, the house, and doesn't need much of the space. And as we know, chicken just grows so fast every few weeks. So you can, so really a generation interval, just 20 weeks age, the chicken start laying eggs and then can have another generation. And then, so this product is small enough so the whole family can enjoy it. So it's can frequently available for the consumption. And then chicken, it's people use chicken as a pet. So the really culture acceptable nearly all of those culture. And more importantly, particularly in African country, we need to empower women. So actually for across all of those farm animal livestock, so women is the primary owner of those poultry, particularly in African countries. So really the poultry production contribute to poverty alleviation and women's empowerment and nutritional security. So why we focus on heat stress and Newcastle disease, I don't need to talk about heat stress because all of us probably experience a huge stress from the heat in the past many years, particularly this year. I guess many of you guys experience on that, how important of the heat stress can impact the productivity and the immune capacities. And Newcastle disease, it's indeed is a number one of the disease contribute to particularly the poultry production in the African countries. So without the vaccination can cause 80 to 100% of the bird die for those high pathogenic and Newcastle disease virus, because it's a respiratory disease virus like the COVID-19, right? So they spread very, very quickly between the different flock, different village. It's very, very difficult, very challenging for the African smallholder farmers. So people will question why we're not vaccinated. So in this rural area, so this is very challenging. So the power, it's a major issue in those African countries. So the cold chain, it's a huge challenge. And many of the time, those vaccine could be a fake vaccine because it doesn't have any indication if the vaccine is still valid. And then have a very limited veteran service and extension for vaccination. And then just like the COVID-19, right? I guess many of you guys have a multiple vaccination. It's the same thing for the Newcastle disease, not just a one-time shot will be provide protection forever, need many times. For those scavenging system, it's very difficult to get those birds vaccinated, even have enough of the service. So really, these the two factors that really contribute to the major losses and the decrease of the production among the affected poultry flocks. So this is the program really have 3 different phases and 2 major areas. One is obviously very important on the research, and another area is the capacity building. So for the research, you have 3 different phases. The first phase is called a discovery phase. Basically, we try to identify genetic markers, genetic variants associated with those histories and disease resistance. And then in the development phase, basically, we try to develop that sequencing platform, so economic, affordable for sequencing platform, so we can make a genomic sequencing and improve naturally resistant, enhance the resistance to the Newcastle disease, more resilient of birds. At the same time, we want to conduct a called value chain assessment to see where the niche market and develop a business model for the breeding and the distribution of the resilient chicken line. So the genetic enhanced birds can deliver to the smallholder farmers. Obviously, last phase, we need to do the implementation plan to perform those genetic selection and the breeding and monitoring to see if they are effective for improve resilience. And then also we need to training the breeders and farmers so how they can use that genetic platform so it can be more sustainable in the African countries. And around probably 30 to 40% of our effort actually go to the capacity building, because we need to build the research facilities, the animal facilities, and more importantly, to training the people in the African country so they can needing the more effort in the future so it can be the more sustainable. So here is some of the major outputs I'd like to share with you guys from this program. So basically, we are able to develop this genomic selection platform, and so can improve primary, for example, survival time to Newcastle disease infection, and at the same time can improve egg production and the growth rate. And then we are able to establish those breeding flock for the 2 population from each country in Ghana and Tanzania, and I expected to have 10 to 15% of improvement, for example, the survival time. And then we are able to develop this business model for the breeding and marketing for the genetic improved lines. And then we really, in the past 10 years, we really engage with the private sectors. So in the future, how we can collaborate with them so they can deliver the genetic line to the smallholder farmers. And then, more importantly, also, we are able to build those poultry breeding station, research facility, diagnosis laboratory. So this is a really critical infrastructure so they can run in those experiment to make a genetic improvement for more of a resilient of birds. And those are the infrastructure as a methodology we developed in the past 10 years really provide a sustainable genetic improved program for the indigenous birds working in the African countries. And as I mentioned, we provide those training to generate a cattle of those poultry professionals so they can have those expertise and skills to leading the research project, focus on poultry genetic and disease. So here I'd like to provide this is a summary of those metrics in the past 10 years. So about how many graduate students we trained through this program, and how many smallholder farmers we engaged, and then how many new technology generated from this program, and the diagnosis laboratory generated partnership, and poultry research facilities, and outreach activities, and publication, obviously, and how many people through the program trained. So the program officially ended last September. As 10 years, it's a hard stop for the USAID program. So very fortunately, just a couple of weeks ago, USAID informed us they will give us another 5 years, $5 million to finish last mile of this program. So hopefully in the next few years, we can have a significant impact to the smallholder farmers in the African countries. So now I can switch to another topic, switch from a chicken, now move to a broad of the farm animal species. So obviously I put the chicken in the top again, so I don't want to offend my colleagues working in the pig and the cattle. So this is one for my graduate student. So she's very talented to make a jaw of this chicken, pig, and a cattle. It's by size, even I'm a bias from chicken. So basically, so this initially for a few years ago, so following actually a path of the human called ENCODE project, basically try to understanding there's a non-coding region of the genome to see what is the functional rule of those non-coding region. We saw that it's a junk DNA many years ago. So start with these few of the core tissues, and basically to try to employ a variety of the epigenomic assays to understanding. So first identify where are those regulatory elements, what is the function of these across different tissues, and provided this resource to the animal genome community. So we are able to publish this work few years ago for this type of work. Specifically, why we care about the functional application of the animal genome. I know many of you guys work on animal genetics. So no, this is a really challenge to identify the causal variants for the economically important traits. We know there's so many of the genetic variants can contribute to the complex traits. And it's hard to find those, where are those causal variants. And then based on many of those studies, we know the genome-wide association analysis is a typical method of how you can identify genetic variants associated. So not the causal to the phenotypic variations. So 90%, more than 9% of those genetic variants in the non-coding region. So that's very important to understanding those non-coding regions. So we know the functional rule of those non-coding variants are context dependent. So later on we'll talk about why of this is important. So it's not like just look at the DNA information, because the same DNA sequence could have a different functional meaning in different contexts. Depends different tissues, different development stage, different physiological status, different nutrition treatment. So that's how this the annotation is such a fundamental important. Obviously, so without those information, the first step it needed to develop of the comprehensive catalog of the functional elements in the farm animal species. So we can, once we're learning of those causal variants, we can improve genomic selection accuracy of the breeding. And then as a biologist, we always try understanding the biology behind these. So what are target genes of those causal variants? What is the molecular function of those causal variants? So based on the past of the many of the study, we know the variation in the gene expression are the major factor accounting for phenotypic variations associated with the complex traits. If you think about, start with the single cells, right? So all of the DNA, if you compare the DNA sequence from those single cell, then to the nature of the different tissues, you will see their DNA sequence, DNA coding nearly exactly the same. But when those single cells start developing into variety of different tissues and organs and have a different biological function, right? If you will see hundred of the genes, so there will be expressed in the different level. So even with the same DNA sequences, right, indicated those context dependent of the functional information is so important, and the gene expression of the gene expression regulation is playing such on the rules contribute to the complex traits. So this is a model developed by a professor in Stanford University. So talk about how those genetic variants can contribute to the complex traits. If you look at it here, so primary through the chromatin, which through the transcription factor binding, and then histone modification, so can directly affect the messaging RNA. And then obviously they can through the splicing and have a direct impact for the gene expression then contribute to the complex traits. So I like to use this flow chart to demonstrate how that important of the functional information of the regular elements. So this is the typical of the work called genome-wide association analysis as a Manhattan plot. So you will see lots of the dots here. This is obviously the highest one, which means they are significant associated with complex traits. But each of those genetic variants, if you look at it here, that's what we talk about the functional information. So if you look at it here across many different tissues, so from here you can see it depends where are the SNPs, genetic variants located. If they have the peaks, depends which of the tissues. If they have the peaks from here, you can see like for bone, for these two genetic variants. So which means these genetic variants in this position, they are functionally related to the bone-related biological traits, right? Even many of those SNPs you identified, if you are doing the genome-wide association analysis for the bone-related traits, so many other SNPs you identify may not relevant, but only these two SNPs, genetic variants, potentially are causal variants. So you can narrow down to these variants to doing a functional analysis to validate if this is a causal variance for the breeding and selection. And so here, that's how I like, so give the idea about, so when we do the genome-wide association analysis, we're talking about getting the DNA information and phenotypic information, and we're doing the association analysis. That's really one dimension of the information, right? So now we talk about it because those genetic variants, it's context dependent. If you think about these, basically it's talk about here, just you talk about three different development stage, and each of the development stage have different tissues depending on which of the biological traits you are interested. And then we haven't talked about physiological status like infection, nutrition, innovation. So you can see this is a really multi-dimension of this. Plus, you need to have many of different epigenomic functional assays. So, and then it's really come to a multi-dimension of these. So we really want to move to this, apply different functional assay. And so this is a different assay as basically not any single assay we're able to characterize of this functional information. So that's we work on the chicken as across all of the 23 tissues. And this is the work probably last year. Basically from here, you can see, so we can characterize 5 different promoters, 5 different enhancers, and they use a combination of the different assays. And then here, just to give the one of the example, so you the modern broilers, this is the COP 500, and then this random mating, you can see if you look at the body weight, they have a significant difference between their body weight. And then if we look at the epigenomic assays of the COP and random mating of those meat-type of birds, they all have those strong promoters. But if you look at the enhancer, you can see only COP have an enhancer, no enhancer from random mating of the meat-type birds. And then we look at this specific enhancer region, you can see there's a 2 allele specifically. It's this allele from ACRB, and this one only for the COP. So they are really diverse, the allele. And then if you look at it here, 3-dimensional structure, so you can see this is a single nucleotide change, can have here have the chromatin interaction versus here doesn't have interaction. Point a potential causal variance of these potential causal variance contribute to the significant difference in terms of the COP 500 and a random mating. And this is in peak, basically utilize like 50 to 90,000 of those peak in 3 different breeds. And again, look at those promoter enhancer in the tissue specific manner. You can see they're highly related to the muscle, adipose tissue, gut tissue, and brain tissues, which is biologically relevant to the average DNA gain. So here, just to give the example, how we can utilize the genome-wide association analysis, narrow down to those enhancer regions, and then utilize the 3-dimensional structure to identify targeted genes if you look at this here, the ALPK2 really highly expressed in the muscle, not in any other tissues. And the meaning of this potential ALPK2 is, biologically important of the genes contribute to the average DNA gain. So this is the switch to the cattle function annotation of the bovine genome. In that case, it's talk about the different development stage. So if you look at the expression of this, you can see between the fetal and adult muscle only have a few hundreds overlap. Most of them are totally different. If you look at their marks, you can see in the fetal tissue, they have many of those enhancers, which the adult tissue just have a very one of these. If you look at their annotation, you can see overwhelming of all of those enhancers, but this is a repressive marker in the adult tissues. And then another stuff is machine learning, how we can, use AI so we can predict of the function of the DNA sequencing, give the input of those sequence information, and how we can learning what is the functional rule of this. There are regulatory and functional regulatory elements, or how important of those functional rule of this. So, and then another thing is when for some of the tissues, so we are able to get those assay workout, we can impute it of this. This is the red color of these, many of these we're not able to work out. So we can really, through those imputation, and then you can see accuracy, it's very high, even we don't have those assay available. So last, I like to touch about kill bone damage. So this is really portrait welfare issues. So if you look at here, so this is the happened in the commercial layers. The study indicated 50 to 98% of these have these occurrences. And then the genetics and nutrition and housing system play a very important roles for the kill bone damage. So again, this is a really nice multidisciplinary research team. So we work together. And then the major objective is to identify genetic markers for the kill bone damage to reduce the occurrence of the kill bone damage, evaluate the potential house design innovations, and pinpoint a correlation of the behavior or production related kill bone risk, assess and predict of the economic trade-off industry and the consumer returns of the improved of genetic resistance and the house innovation, and establish of the kill bone damage of early detection system by using genetic and behavior of data. Really, it's innovated, and so we focus on interactive of the effect of genetic, layer, environment, and behavior individual contributor factor to assess, test, and evaluate the feasibility of the house design innovations, provide economic of the analysis of trade-off genetic and house innovations, to guide individualized and industry-wide of the solutions to determine industry consumer social return by examining of the impacts of the egg industry, which is the supply side, and egg markets, which is the demand side, and then develop of the precision livestock system, and for early detection of the kill bone damage, use the data and an opinion of the evaluation. So some of the perspective, this is the economic important traits improvement, particularly these resistance, really challenge to improve. Really context dependent on the multidimensional function, we can really accelerate of these identify causative genetic variants for complex traits. Genetic diversity and adaptation of the local environment are really important for the sustainable genetic improvement. And I really believe a holistic approach with a cross-discipline of knowledge will be essential to improve the animal production. And then we really need to take advantage of lots of cutting-edge technology available for us. I do believe it's a great and right time to bridge the genome and a phenome gap. I really like to thank my group and funding, and then many of the collaborators in the world. I really truly believe collaboration is everything. So I did enjoy the Kiole Hall. That's how I visit Kiole yesterday, and chicken, I stand behind the chicken. So and the whole family enjoyed the beautiful campus. And then once I'm a cyclone, I'm always a cyclone. Thanks very much for your attention. Sorry, I'm still over time.
Give him more time and he's still not doing it. Yeah, so there's no questions, but yeah, you'll be around. So yeah, appreciate the talk. Very a lot of work, obviously, and a lot of collaboration. So appreciate that. We should have played the chicken dance, right? Yeah. But we'll have a dance tonight. The dance floor is already there. Yeah. And Huaijun, you're going to lead the chicken dance. Yeah.